Thank you for that introduction. Today, I'm going to be talking about applying omics approaches, bioinformatics, and machine learning to studying the companion animal microbiome and the human microbiome. Now, many of you are going to be intimately familiar with this topic, but hopefully there's something new for everybody here. So first, I'm going to work through the definitions of what the microbiota and the microbiome are. We'll talk about some functions of the microbiome, and then how omics, bioinformatics, and machine learning are used synergistically to study these complex bacterial communities. And then I want to go through some examples that I've selected where researchers have used these techniques to study the microbiome to learn interesting things about the community or how they're relevant to health and nutrition. And then we'll do a little teaser about ongoing work at Nestle Purina. So first, the microbiota is typically defined as all the microorganisms that live in a reasonably defined habitat. This includes bacteria, archaea, fungi, protists, and controversially, some people include viruses or phages in that definition. But in the 1980s, some researchers working on the rhizosphere, which is essentially the microbiome at the interface of plant roots and soil, came up with a nice definition of what a microbiome was that I like very much. They defined the microbiome essentially as the microbiota plus its theater of activity. This includes all of their genomes, all of the transcripts of their genes. It includes the extracellular material they have, plasmids, gene cassettes. It also includes their metabolism and the products of that metabolism, so their consumption of different molecules and everything that they produce. It also includes all the physical and chemical properties of the environment in which they reside, and all the interactions between the members of that community and between the community and the environment, or the community and the host. A couple of reasons I really like this definition is that it really links the idea of a microbiome to the ecological idea of a biome more generally, which is typically defined as a geographic area that's defined by its characteristics and the organisms that live within that. So you can think of like tundras and forests. And then the other reason, sorry, I'm going to go back real quick. The other reason I really like it is that it introduces us to many of the omics that we use to study the microbiome. The genomes and their transcripts, that's metagenomics, that's metatranscriptomics. Metabolism, we have metabolomics, all the proteins that they produce, metaproteomics. So it's a definition that kind of leads you very nicely into the tools that we use to study these communities. Now, animals host many microbiomes across a wide variety of their body habitats. Many of these are really well known, really well studied. You will all recognize the gut microbiome most likely, the vaginal microbiome, the microbiome of the mouth, the respiratory system, the skin. But some of these are really emerging areas of research that only in the last decade or two have kind of taken off. So for instance, the urinary microbiome was once a question mark. Urine used to be thought to be essentially sterile, but over the last 10 to 15 years, largely as a result of some of the technological advances that have been made during that time, it's now pretty much an accepted fact that the bladder and the remainder of the lower urinary tract house their own microbiomes. And recent evidence suggests that the kidney itself does as well. And I just saw in the last 6 months or so, there's this question arising in the literature as to whether or not the brain itself also hosts a microbiome. This is an open question. There's evidence of it in fish, but whether or not it exists in other vertebrates, we don't know yet. Microbiomes and their microbes perform many functions that influence host health. If you look at just the gut microbiome, for instance, they can inhibit pathogens through direct competition or producing antimicrobial compounds. If any of you are looking for a fun way to spend 5 minutes online later, I encourage you to look up type 6 secretion systems. See if you can find a video of bacteria stabbing each other with these poison-tipped spears that they produce out of their own proteins. They also produce or modulate the production of key metabolites that are important to health and nutrition, such as producing short-chain fatty acids, which are a primary energy source for the cells of the intestines. They can degrade or modify dietary and host molecules. So carbohydrates that we or animals consume as fiber are often not available to the host until microbes actually degrade them. They can also influence host structure and function directly, so through stimulating mucin production in the gut and directly influencing the integrity of that gut barrier or influencing inflammation profiles. Because they have so many functions and they produce so many things that are important to the host, they dramatically influence health, both within the gut itself, but also through circulating. Some of those metabolites go circulating, they affect host signaling, they have effects across physiological systems, across the entire animal. And that has led to the idea of what we call these health axes. So you have a gut immune axis, a gut brain axis, where the functions of the microbiome in the gut are actually influencing the brain and behavior or skin and allergy. So they have these broad ranging effects. And this is why we study them. A very quick look through PubMed or Google will find that microbiome has been associated with pretty much any disease you want to pick, right? So type 2 diabetes and obesity are key examples that have been worked on for years. Kidney diseases, the development of urinary stones, predisposition to cancers, and allergy and atopic dermatitis. There are so many of these. Much of the evidence for these associations is coming from correlative studies, using these kinds of omic technologies that we use now, often done on prospective or retrospective cohorts, and a lot of that employs machine learning. There's, for some of these, also a good causative set of evidence where it's coming from clinical trials, animal models, or in vitro assays. Now, meta-omics, and I'm going to describe what that is in a second, really arose as culture-independent solutions to studying microbiomes. In the 90s, there was a seminal paper published that basically identified what the authors called the failure of microbiologists to describe natural diversity. And the idea arose that less than 1% of the total microbial diversity in the world was either cultured at the time or could even be possibly cultured. And this has really been a pervasive idea in the field ever since. Their proposed solution was to use the 16S rRNA gene, which had been worked on for about 20 years at that point, as a kind of barcode that would allow you to identify microbes, as a way to study an entire community as a whole. So what you would do is you would take all the 16S rRNA genes, amplify them, clone them, and then quantify them, query them against databases of 16S genes from isolates that you knew you could identify. And that's how you would identify and quantify what was in a community. Now, this allowed you to do a couple things. You could measure the whole community as a whole, and this is where the meta-omics comes in. Meta in front of one of these omics basically means we're taking everything at once and we don't know who's producing it until we look at it later. And it allows you to study the community in situ. You don't have to take it out, grow it somewhere else, and introduce more experimental biases. And it creates this opportunity to do things in a high throughput way, to develop public databases, and to keep building tools. Since then, that has led to where we are now in an omics revolution where there's a wild diversification of things that we can do. So now there's an increase in moving towards whole genome metagenomics instead of 16S, metabolomics, metaproteomics, metatranscriptomics, and at even higher throughput through advances. And I would even point out that some of those advances are leading to a revolution that's going on in those culture-based methods. And I'm going to talk a little bit more about that later. This is really exciting. So a typical meta-omics workflow looks like this. And I went through this slide last night and realized this is a lot of words. So let's go to the right side. The whole goal of the thing here is to identify new biomarkers. Those could be microbes themselves. They could be genes or pathways. It could be proteins. It could be metabolites that are produced or modulated by the microbes. It could be looking for targets to use as new biotic solutions, new probiotics, new prebiotics, new postbiotics. Or it could be that you want to develop a predictive model or an ML model. We heard a distinction about that earlier, to use as a diagnostic, so essentially to build a tool. Now, the typical workflow starts with samples or data in a public repository that begins at one of these very complex machines, a sequencer, a spectrometer, or you're performing chromatography, and then it goes into bioinformatic processing step. Now, this is basically where you turn the gobbledygook that comes off of the machine into interpretable biological data. And that step also uses a lot of ML at this stage. It's where quality control happens to the data, and it's where you're querying public databases to see what you can identify. The result of all that bioinformatic processing is typically a feature table. So This is where we're getting that high dimensional data where you have hundreds or even thousands of features that are measured on however many samples that you have. And then the purple box, which is the one where I usually live and is my favorite part, is where you're actually analyzing the data, developing models to try to draw inferences about what happened in your experiment or to draw associations. You'll see ML all over this slide, and that's because machine learning has really been integrated into all of these steps at this point. So it can be used for feature reduction, clustering, or developing these predictive models. If I knew more what was happening inside these complicated machines, if I were more of a technologist, it's probably there too, and I could tell you what it was doing, but we're just going to assume that for the moment. So we've heard a little bit about the distinction between predictive modeling and statistics and machine learning, but I think there's a complementarity that happens with these methods. So most of the statistical methods that you use to study the microbiome are really focused at inferring associations from your sample data, and they're really rooted in hypothesis testing and estimating parameters. They're highly interpretable. They're often descriptive. Many of the things that you do in those statistics are actually rooted in the domain theory, so they might actually come from ecology or population genetics or the field where they were developed, so they have a biological interpretation already. Whereas in machine learning, the focus is really typically on building a tool. And you're using that tool because of its predictive ability. So algorithms that you build in machine learning typically learn from a training set, as we heard earlier, and then they're used to generalize the predictions that they learn how to do with that training set into a test set. And then the key features that are responsible for those predictions are identified by how important they were to the ability of the algorithm to make the prediction. They excel at working with high dimensional data. They're very good at combining different types of data, but they do suffer from a little bit of a black box problem. Some of these machine learning algorithms can be very hard to interpret. So you don't necessarily know why a particular feature was picked. You don't necessarily know why a particular prediction was made. This isn't true of all these algorithms, but many of them do suffer from this. So now I want to go through some examples of people using omics and machine learning and some of these methods to learn interesting things about companion animal microbiomes, human microbiomes, and how they influence health and nutrition. So a recent study here made a massive bioinformatic push across 14 studies, both published and new, to basically identify that dog, cat, and human microbiomes are distinct communities that share some features. We already kind of knew this, but I think there were some interesting results that came out of this that surprised me, and I think are really interesting. Plus, they did something here that helps us all, and this is one of the key features, I think, of this omics revolution. From this giant data set that they assembled from published and new studies, they pulled out almost 30,000 metagenome assembled genomes. These are essentially draft genomes of bacteria that weren't in a database before, and now they are, and now we can all interrogate them. So what they found was that 73% of the bacteria that they were able to identify were host-specific. They were only found in either cat, dog, human. But I think the really interesting result here is that for the bacteria that they were able to identify that showed up in more than one host, there was a range of variability in how specific the strains of those bacteria were to the different hosts. So some species like, this used to be called Bacteroides, and now they've made it unpronounceable. Fascicola, I'm going to go with Fascicola vulgaris, were really genetically similar, meaning that there wasn't really much differentiation in their genomes across hosts. But for other bacteria like Blauschia wexleri, they found that these bacteria have very, very specific cat strains, dog strains, and human strains in clusters. And that's a really interesting result. And I want to point out that this is becoming more and more common, this pulling mags out of a study like this and putting them into a database, either public or proprietary, so that really this combination of omics and big data is just fueling the development of more tools for us. So we haven't really dived into this yet, but machine learning algorithms can be supervised or unsupervised. Here's an example of researchers using an unsupervised method, which is just a fancy way of saying we didn't know anything about the samples ahead of time, right? To essentially cluster microbiomes by how similar or different they are. So you have no subject information. Your real question is, how many clusters can I identify? How different are they? Can I take this big n-dimensional thing and reduce the number of features that I want to look at? This is typically done in an exploratory way, and then the results are fed into another analytical framework. But what these researchers did was that they took 3 very deeply phenotyped cohorts from Dutch studies, yielding, I think, 4,000 people, looked at 16S sequencing to identify the relative abundances of bacterial taxa, and they did clustering on each one of those independently. Were able to find 2 clusters in each group, and then they fed those into basic statistics, right? So where they were taking all those deeply phenotyped profiles that they had and associating them with the clusters. And what they found was that there was essentially, in each one of these studies, an unhealthy cluster and a healthy cluster. So metabolic, lifestyle, and other physical metrics were very, very tightly linked to where these clusters were. And then they were able to identify which bacteria defined those clusters. In a supervised machine learning algorithm, you're doing kind of more what we've been talking about with machine learning, which is that you have a cohort where you know the answers that you want to predict. So you divide your data into a test set and a training set. You create an algorithm based on the training set, and then you apply it to the test set. So this paper from 2022 basically uses this approach to identify that the microbiome can be used to predict chronic enteropathy very, very, very well. This is a very, very common approach using machine learning in microbiota. This is one of my favorite examples. So another thing that you can do is predict responses. So you're not necessarily trying to assign a category to an individual, but in this case, it's a nutritional response. So Coram et al. In 2017 did an interesting crossover experiment where they fed either sourdough bread or commercial white bread to 20 people for week-long periods. And what they found using kind of traditional microbiome analysis methods and basic statistics were there were no bread type-based significant clinical markers that jumped out. Right? So it didn't affect glycemic responses or anything, sourdough versus white. There were no mean effects. Microbiome wasn't affected by the bread type all that much. Very, very modest effects. But they were able to identify this huge variability in how people responded to the different bread types, which is an aha moment, because when you have that kind of variability, the next step is, let's build a machine learning model, which is exactly what they did. And what they found was that they could actually build a model based on the starting microbiome of these people to predict whether or not they would have a better glycemic response to the sourdough or the white bread. So is sourdough bread better than white bread? I would say yes, but biologically probably it varies. Your mileage will vary, and your microbiome probably has something to say about it. Another thing that you can do, we've heard about a methylome-based age model. So this group, working with a cohort of healthy South Koreans from 20 to 80 years old, actually built a microbiome age model. What they found using kind of those traditional methods was taxa that differed in relative abundance between young and old people. But if you just look at clustering, if you look at typical ordination methods, you didn't really see wild patterns that explained a lot of the variance in the data. But a machine learning algorithm was able to be constructed that would predict the age of these individuals with a mean absolute error of about 5.5 years. Now, I mentioned earlier that machine learning models are very good at combining different data types. The next thing that they did was to do just that. So when you add urine metabolites to this model, the model becomes better. And it's not just adding more explanatory variables, they actually refit based on both. I also mentioned that some of these machine learning models can be kind of a black box. So here's an example of a group that kind of overcame that. They essentially developed a model to predict type 2 diabetes risk, and then from the results of that model, they actually constructed a simple index, and then they demonstrated that that index was correlated with increased risk in different categories, and also correlated with circulating serum metabolites, which is exactly what you'd want to see in this instance. So here at Nestle Purina, we're currently using some of these methods to do several things, but a couple of the key ones I wanted to point out. and again, this is ongoing work, so we can't really talk about the results yet, but watch this space, is to predict metabolite production from microbiome features when you're feeding different ingredients or blends of ingredients. And then also using data from cohorts to associate microbiome features with pet parents' sentiments about their animal's health. I think that's a really interesting thing when you don't have phenotyping. If you can use questionnaire answers, that connects to why someone might bring their animal to see all of you, right? So I think that's also a useful exercise. I also kind of hinted that there is now this ongoing revolution in some of those culture-based methods based on all of the advancements that we're seeing. So this paper, when I saw it, really, really excited me. Traditional methods of developing a culture bank from an individual are really difficult. They rely a lot on serial dilution, where you're taking an arrayed format like 96 well plates or a 384 well plate, and you're basically diluting what you have to the point where you expect monoclonal growth of only a single type of bacterium in each well. When you do that, you can then use random picking to try to isolate a bunch of bacteria, or you can use selective media to try to encourage specific kinds of bacteria to grow in them, and then pick those. But it's very laborious. It tends to isolate the same common organisms over and over and over and over again. So you have to keep doing it, keep doing it, keep doing it in order to get a good variability. And it's really difficult and time consuming and hard. But this group has developed a system they're calling CAMI, which is culturomics by automated microbiome imaging and isolation. This is fascinating to me. So even within an anaerobic chamber, they're able to get this to work, where you take an imaging system that can measure bacterial colonies' shape, size, color, texture, and other features. You pair that with an image-guided machine learning algorithm, and then you pair that to a robot that's performing rapid selection of those colonies. It all goes into a genomic pipeline where you're using omics, DNA sequencing, to identify those bacteria and characterize them. And then it also goes into a biobank, and it fuels the digital database. And what this allows them to do is to make predictions about which genera they think they're picking. Is this overrepresented, underrepresented in their database? What do they want? It allows them to basically say, we've already got this, we don't need it, don't go get it again, right? And those are all ML predictions. So in a test of this, they took 20 people, retrieved 102,000 colonies, 27,000 isolates, almost 1,200 draft genomes recovered from this, and almost 400 16S ASVs. That's a little bit like saying bacterial species. And they're calling this cultured biobanks on demand, which is really exciting. So in summary, the examples that I've selected, I really wanted to demonstrate that the synergies between these methods are helping drive advances in microbiome research and how it influences health and nutrition. So on research advanced sides, these are opening up new systems approaches that take multiple omics into account, looking at multiple systems within the host. They're expanding databases, they're expanding the availability of public pipelines for researchers to use. And on the health advances, we're moving towards getting novel predictive diagnostics, age diagnostics, type 2 diabetes diagnostics, and new biomarkers are being identified. Novel biotics are being identified, and we're increasing our ability to make personalized, precise measures about health status and nutrition. All right. So several people have helped with all the ongoing work, and I'd love to thank all of them. If you're not on the list, I apologize. All right. Thank you very much.
I want to take a moment to remind everyone that you could submit your questions by Slido, or you can go to the microphones and we'll alternate between them. And we have a couple questions for you.
Okay.
Yeah. So how do you determine if an organ has a microbiome, kidney or brain, for example? Does it simply mean that there are microbial products that are found in the organ, or does the organ have to at least have genes for microorganisms to state that that organ has a microbiome?
So I would say that the typical answer to that question is that you need to define that there's actually a persistent community of microbes that resides in the organ independent of transient infection, and that community exhibits some measure of stability over time so that you can keep recovering it. That's, I think, really the measure. Like, there has to be a stable, persistent community that can't be just explained by infection.
Is there such a thing as a healthy, in quotes, microbiome, or are we moving much toward a functional microbiome model?
I think if you asked everyone in the room, you'd get a different answer to that. So there are probably lots and lots and lots of ways to have a healthy microbiome. We know a lot of the features of a healthy microbiome. They don't necessarily mean that every healthy individual has the same taxa all the time. There's going to be variability between people. We just know that's a fact. Now, to the second part of the question, I think we are getting towards more functional definitions, and part of that is really fueled by better tools that we have now. So in the 1970s, we really had the original development of the 16S rRNA gene, and that was kind of it. It had taxonomic information, and that was pretty much it. As that has moved forward, we now can look at their products, we can study their interactions with the host more thoroughly. We're moving towards function.
So for most of us are veterinarians here of all different specialties, et cetera. What you've presented is mind-blowing. Right? What does it mean? When will it be on our countertops in the labs part of our clinics? Will it be in foods? Like, where is this headed for us that practice medicine every day?
So some of it already is in your foods, right? So if you think about biotic solutions, those are your probiotics, postbiotics, prebiotics. A lot of those are actually informed by this research or coming directly out of it. And then some of the diagnostics, I think, are going to run into, they're going to be developed further as the regulatory space catches up, as the commercial space catches up.
Excellent, and you're up.
Oh, thank you. Thank you for your presentation. In the slide where you mentioned the human, the cat, and the dog, and talking about publicly accessible banks, do you foresee them starting to include any lifestyle factors? It would be interesting to know, did this individual own a cat? Did they own a dog? Did they own both species? Are they veterinarians and they work primarily with dogs and cats? So it might be interesting to look into some of the lifestyle factors around the microbiome as well.
Absolutely, and that kind of data is typically part of cohort studies. So there have been some studies on just that question. There's some evidence that owning a cat, for instance, does influence your microbiome. There was a study published in the early 2010s that really demonstrated that owning a pet influenced your skin microbiome to a great degree, less so the gut. But there are studies like that. But that kind of data tends to be part of independent cohort studies.
Thank you.
Okay, we have time for just a couple more. Are there specific omics technologies that have been particularly useful for nutrition studies?
All of them, yes. I mean, metabolomics, I think, is one of the most direct ways to do this, right? So I think there is a current recognition in the field that the explanatory power of some of these things becomes greater the closer you move to function. And when you're looking at metabolomics, you're looking at the molecules themselves. Some of those are going to be produced by the microbiome. Some of those are coming from the diet. With our improved understanding of a lot of the pathways involved, we can actually see that some of them are modified by the microbiome itself. So I think metabolomics is one where there's a very, very tight direct link. And when you're looking at metagenomics, when you're looking at metatranscriptomics, you're kind of stepping back to focus on the community itself.
Fantastic. Thank you so much.
All right.
We'll get ready now for our next speakers.
All right.
Thank you.
Thank you.